So this is the BioTek EL406 wash dispense device with a BioStack 4 for bulk dispensing and plate washing. And we're going to learn how to use it today. We're going to start with the peristaltic pump dispensing. So first you turn on the BioTek in the back here. Well, I should say first you sign up on the Faces calendar, then put on your gloves and your lab coat, then turn it on. And it takes a little while for this to initialize. And then when it's ready, if you're using the BioStack, you would turn it on now. It has a switch over here, but we're not going to use the BioStack today for the training. That is good for when you have many plates. If you just have one plate like today, then we'll just load it manually here. Okay. So for the peristaltic pump, that's over here, we need to use a cassette, and the cassettes are in this second drawer. They look like this. There's different sizes. The difference between the different ones is the diameter of the tubing. And we're going to use the 10 microliter one today. And it's called 10 microliters because one rotation of the pump is going to be 10 microliters. So if you wanted to dispense 50 microliters, it would rotate 5 times. So you take this cassette, and first you hold this in your right hand and place it in this holder here. Over here. It clicks in. And then you take these parts and lay it below where this pump is, and first click the right hand one on. It actually just like rests there, and place the left one in the silver device, and then sandwich it closed. There's some tension that you have to apply, or like force, and that's okay. You know, it's about as tight as, I don't know, like if you're tying a hairband on your hair or something. So you're not like pushing super hard, but it is a good push. And then so you can see the tubes are wrapped around the barrel. You're welcome to come look if you want to see. These tubes are basically what the device is doing, pushing them against the barrel that's going to rotate, and that's how the volume is going to get sucked up. So these are actually, it's called the slug volume, the liquid that's actually in these little pieces here on the barrel. And then after you close this, you slide that closed. So this end has a weight on the bottom, and that's really convenient for when you're dispensing from a thing like that, because it just keeps all the tubes together. But sometimes you're dispensing cells, and so you're going to need everything to be sterile. In that case, you can take the tubes out, and then you would be able to sterilize them and put them into your cells. You can, since there are 8 tubes that are going to correspond to 8 sections of rows of the plate, you can place these in different solutions if you wanted to dispense multiple different things at the same time into different rows. But today we're going to be using this weight, and we're going to dispense all of it one liquid. So that's ready. And you can see when we turned it on, the screen came on. Okay, so now we go over to the computer. Back up. So we also have a few things we need to check. For using the peristaltic pump, the waste is going to go into this waste bottle. So before you start, you want to make sure the waste bottle isn't too full. Right now it's like this full, which is fine. If it starts to get like 2/3 full, then you have to dump it out in the sink. and then put a little bit of bleach in there. There is a backup bottle over here. It has a sensor in it. You can actually see the sensor. Okay, well, it's hard to unscrew. Anyway, there's a sensor inside here. So if liquid ever goes from this tube from this tank into this tank, then the sensor will go off and your protocol will freeze and you'll have to start from scratch. So the goal is to have, you know, the waste accumulate here and never start accumulating in the other tank. All right. And okay. So you go over to the computer. This is usually left off. It's usually on, but it's not logged in. And so what you do is log in with your cruise ID. This computer is set so that it won't go to screensaver while you're busy doing a dispensing. And that we did that on purpose so that if somebody logs in with their cruise ID and then someone else takes over the work, if they're not around to enter their password in every time the screensaver came on, then this was a problem when we first installed it. So we've solved that. So we're opening the software called Liquid Handling Control. It's this little icon here. And this is what it looks like when it opens. And you would, first you want to tell it that we've put on the 10 microliter peristaltic cassette. So you click on Tools, Instrument Utilities, and then you click on the Peri pump dispenser, and then under cassette type you click on the 10 microliter just to let it know that that's what we loaded, and then you click send, and that sends that information to the device so it knows and the software knows. And then you have to go here to settings on the top left, and then you click get. That allows the protocol to then know what the current cassette is. So it's this whole sending and getting process every time you change hardware. And then you can click on the open, and this will take you to a window in the C drive, program data, BioTek, liquid handling control, protocols. In this folder are a bunch of protocols that you can modify and use for your experiments. So we can let's use one of my demos. Let's see what this is, demo Peri. Okay. All right, so when you open a protocol, it will have a plate type. So in this case, we're going to do a 384 well plate. This is what a 384 well plate looks like. It's got 16 rows and 24 columns. And then it will have some steps in the protocol. In this case, the steps all start with a P. The P is for Peri, for the peristaltic pump. This device also has a syringe pump and a wash module. So the W steps are for controlling the wash module, P are for the Peri pump, and S are for the syringe pump. So if you want to dispense from the Peri pump and you want to add that step to the protocol, you use the P dispense, not the S dispense, or the W dispense, not to be confused. So our protocol that we have here is it's going to prime the line. It's always good to prime before dispensing because right now there's just air in there. And then it's going to do a dispensing. So for each of these steps, you can double click on it if you want more information. So like the priming, it's going to dispense 700 microliters per tube, just into the waste bucket. And it's going to go at a low flow rate. The flow rate's dependent on what it is that you're dispensing. You can actually, for this, just do high. And you can say what cassette's going to be in there. And then for the dispense step, we're gonna do 10 microliters per well. Let's do 20. 20 microliters per well. Let's do a medium flow rate with the 10 microliter cassette. There are also advanced options. If you click on advanced options, one of them is this pre-dispense option. You'll see when we dispense, it does a little like a quick prime before it goes to dispense, and that helps clear the line of bubbles. So we check that on, and then this is where you can select what columns you want it to dispense to. You know, if you had an experiment where you have one cell type on the left and another cell type on the right, for example, then you might, you know, program it to just dispense half the plate first. And then you can also specify what rows you want dispensed, because this is a 384 well plate, and there's 8 ports with 16 rows. Each port is going to do 2 rows. It's going to do a row on the way down and then another row on the way back. And so that's why your options are, you can do the second row each time, or the first row, or both. Those are your options. So we'll do both. And then on this protocol, I just have some prime steps at the end for cleaning the line after we're done. Let's see. Okay, so once you have your protocol ready, then you want to place your sample in the holder. So this is my microplate, and I'm putting it over here in the holder. And the corner A1, there's a reminder here that A1 should be in the top left. You know, it's very easy to, if you're not looking carefully, put your plate in the wrong way. And that'll be fine if what you're dispensing is even. But, you know, if you care about what's where, then it's always good to just have the A1 in the correct place. Actually, you know what? We can dispense dye, because then we'll be able to see what we're doing for this demo. So in this case, I just have food coloring in some water for demos. And you want to make sure that the tubes are deep in enough in the liquid so that, you know, if it was floating, then, you know, it would just be sucking air. So you want to make sure they're actually like in there. Yeah. So the plate's on, the liquid's ready, so now we just click run. And then it just asked me, it often does this, do you want to save changes to the current protocol before proceeding? So I made some modifications. It's always good to kind of think first, do you want to save those changes or not? If you say no, it'll just continue and it's fine, but you know, you can save it later. But if you say yes, then it's just gonna do a save. It's not a save as. So if you just modified somebody else's protocol, for example, then you definitely don't want to overwrite theirs, you know, before renaming it. So you could say no and then do a save as rather. In my case, I actually like my new settings, so I'm going to say yes. And so it's basically going to just go through all the steps. This is the prime step it's busy doing. That was the pre-dispense. Now it's doing the first set of rows and the second set of rows on the way back. And that's it. That was the dispensing. And then on this protocol, I have some wash steps, because obviously we can't leave the cassette like this when we're done. We need to clean it for the next user. And so I have first a step that says we're gonna put soapy water. And it's just a priming for 5 seconds. So that got some soapy water through there just to get that food coloring out. And then next we'll do some DI water. I have this programmed so that there's delays in between the steps so that I have a moment to switch out where the tube is. But if you're getting really good at it, you can delete those delays and just move from one to the other. It's always good to make sure you're cleaning the outside of the tube. This is a good example. So see how it's soapy there? That's because I only just dipped it in the water like that far, so the soap from the previous step is still there. When you're doing washing, you always want to make sure you actually wash the outside and the inside. The priming is only doing the inside. You have to kind of like manually rinse off the outside. And this is where you can see this is the waste bucket that connects to a vacuum pump, and the waste ends up going to this waste bottle down here that I was talking about. That's what that's for. And then I'm just going to do a final rinse in clean water. Just because a little bit of soap and a little bit of green food coloring may have made its way into my flask just from the transfers. So now I have some very clean water for a final rinse. And then the last prime step, I just pull the tubes up a little bit so that now the intake is in the air. And so that way when it does the last prime, it's basically drying out the tube. And so now it says the run has completed successfully. So it worked. We got through all the steps, and now we need to put away the cassette because we're done. So you would pull this back, and then to release the cassette, you push this little lever here, like that. It kind of unsnaps. So I can show that again. It goes like that. And then you just gently move it over and take it out. If you were coming back to dispense something else very soon, then instead of taking it off, you could put it in, there's like a intermediate holding pattern. There's a little lever that you pull out here. And then that way when you release it, it actually just sits up like that. This is a good position to be able to leave the tubes and they're not under tension anymore. So this is like a good way to let it wait if you're going to use it again soon. But don't leave it overnight like this. If it's an overnight deal, then just put it away. In which case then, if you come and it looks like this, you just push this away and then it's able to open. And you can gently slide these out and pull this up. And that's your cassette. To put the cassette in its holder, the easiest is just to start with the part that has the writing on it. And there's a cutout piece that fits just that part perfectly. So you place that one in, and then from there it's pretty easy to see where that side goes. When you place this one, you don't want to put these tubes under tension. Basically all of this is to make it not be under tension. If I put it there, now it's under tension. This would be bad for the tubes. So I'm just giving it like a little bit of slack. But you also don't want to put it there, like this is now, it's like reverse tension, it's like scrunched. So you just find a position that's like relaxed, and then the rest of the tubes coil around there, and it's now ready to store. It does have a little bit of liquid in, but that's okay. It's kind of unavoidable. And you just put the lid back on and put it back in the drawer for the next user. And so this is the plate that we dispensed. Lovely. Looks pretty even. So imagine hand pipetting this. You wouldn't probably have been able to get it as even. There's actually not even any bubbles. This is a very good device. So So we're done. What you would do next is turn it off in the back, and for the computer you would close the software with the X, and then you would click on the start and go to your little profile icon and say sign out. That way nobody else can get into your cruise ID, because like I said, there's no screensaver, so it's never going to, you know, prompt you to re-log in. It's just going to then forever be logged in until someone logs you out. And you can just leave this on when you leave. You don't have to turn the computer off. And so that was the BioTek peristaltic dispensing.